So here's something worth thinking about as you're settling in tonight. Right now, at this exact moment, there are viruses everywhere around you. On the pillow under your head, on the blanket you just pulled up, drifting in the air you're breathing, sitting on your skin. And before that sounds alarming, here's the thing. The overwhelming majority of them can't do anything to you. They're not yours to worry about. Most viruses on Earth infect bacteria, not people. Some infect plants. Some infect insects or fish or fungi. The ones that actually target human cells are a tiny, tiny fraction of the viral world. But that tiny fraction has shaped the course of human history more profoundly than almost any army, any empire, any invention. Viruses have toppled civilizations. They've decided wars. They've driven the evolution of every living thing on this planet, including you. And the wildest part, the part that still makes biologists shake their heads, is that we didn't even know viruses existed until about 130 years ago. For almost all of human history, we lived alongside them, died because of them, and had absolutely no idea what they were. And that's what we're going to explore tonight. Viruses. The whole strange, fascinating, sometimes terrifying, often beautiful story. We're going to start at the very beginning, long before anyone knew what a virus was, back when plagues seemed like acts of God or punishment from the heavens. And we're going to work our way forward through the centuries, through the brilliant scientists who slowly pulled back the curtain, through the pandemics that changed everything, all the way to the astonishing things we're learning right now. We'll take our time. There's no rush at all. If you drift off somewhere along the way, that's perfectly fine. Some of the best ideas come to you on the edge of sleep anyway, when your mind is loose and not trying too hard. Let's start with what people used to think about disease, because that context matters. For most of recorded history, sickness was a mystery. The ancient Greeks thought illness came from imbalances in the body's four humors: blood, phlegm, yellow bile, black bile. Get them out of balance, and you get sick. The treatment? Restore the balance. Bloodletting, purging, special diets. It sounds crude now, but the humoral theory was remarkably persistent. It dominated Western medicine for nearly 2,000 years, from Hippocrates in the 4th century BCE all the way into the 1800s. Think about that. The same basic framework for understanding disease lasted longer than the Roman Empire, and alongside the humoral theory there was another idea, much vaguer, much harder to pin down, called miasma theory. The idea was that disease came from bad air, miasma, foul-smelling vapors rising from rotting organic matter, from swamps, from sewage, from the dead. If a neighborhood smelled terrible, that was considered dangerous, not just unpleasant, but literally sickening. And here's what's funny about miasma theory. It was wrong about the mechanism, but it accidentally got some things right. Places that smelled bad often did breed disease, not because of the smell itself, but because the same conditions that produce bad smells, stagnant water, decaying waste, poor sanitation, also happen to be the conditions that breed the actual agents of disease, whether those are bacteria or mosquitoes carrying parasites. So cleaning up filth and improving sanitation, which miasma theory encouraged, actually did reduce disease, even though the reasoning was completely wrong. Sometimes you can do the right thing for the wrong reason, and it still works. But here's what nobody had, for centuries and centuries: any concept of a specific, tiny, invisible agent that caused a specific disease. The idea that sickness could be transmitted by something too small to see, something that moved from person to person, something alive or at least active. That idea was slow to develop. There were hints, though. In the 14th century, during the Black Death, People noticed that plague spread from person to person. Quarantine practices developed. The word quarantine itself comes from the Italian 40 giorni, meaning 40 days, the period that ships arriving in Venice during plague outbreaks had to wait before passengers could come ashore. So there was a practical understanding that isolation could prevent spread, even if nobody knew exactly what was spreading. And then, in the 16th century, an Italian physician named Girolamo Fracastoro proposed something genuinely ahead of its time. In 1546, he published a book called De Contagione, in which he argued that epidemic diseases were caused by tiny seed-like entities he called seminaria, which could be transmitted by direct contact through contaminated objects or even through the air at a distance. That's remarkably close to the modern understanding. But Fracastoro couldn't prove it. He had no microscopes, no way to see these hypothetical seeds, no experimental framework to test the idea. It was a brilliant intuition that the world wasn't ready for, and it largely faded into the background as miasma theory continued to dominate. Now, there's another thread running through this early period that's worth mentioning, because it connects to where viruses enter the story. Throughout the centuries, people noticed that surviving certain diseases seemed to protect you from getting them again. This was especially obvious with smallpox, one of the most devastating diseases in human history. People who survived smallpox, those who lived through the fever, the pustules, the agonizing illness, almost never got it a second time. Their bodies had learned something. They'd acquired some kind of protection. Nobody understood how or why, but the observation was clear and consistent. And in parts of Asia and the Middle East, people actually tried to exploit this by deliberately exposing healthy people to mild smallpox material, a practice called variolation. They'd take dried scab material from a mild case, scratch it into the skin of a healthy person, and hope for a mild infection that would confer lifelong protection. It worked, mostly, but it was dangerous. Sometimes the inoculated person developed full-blown smallpox and died. It was a gamble, and the stakes were your life. But the fact that people were willing to take that gamble tells you something about how terrified they were of the disease itself. The real turning point, the moment when the invisible world began to reveal itself, came in the late 1600s, and it came through a lens. Antony van Leeuwenhoek, a Dutch draper and self-taught lens grinder from Delft, built microscopes of extraordinary quality, single-lens instruments that could magnify objects up to about 270 times their actual size. And in the 1670s, peering through these lenses at drops of pond water, at scrapings from his own teeth, at all manner of biological material, Leeuwenhoek saw something no human being had ever seen before. He saw microorganisms, tiny living things swimming around, clearly alive, clearly real. He called them animalcules. He wrote detailed letters to the Royal Society in London describing what he saw, complete with drawings, and the scientific world was stunned. There was an entire invisible kingdom of living things, right under everyone's noses, and nobody had known. Now, Leeuwenhoek didn't make the connection between these tiny organisms and disease. That would take another 200 years. But he opened the door. He proved that the invisible world existed. And eventually, in the second half of the 19th century, two towering figures would walk through that door and transform medicine forever. Their names were Louis Pasteur and Robert Koch, and between them, they established the germ theory of disease, the idea that specific microorganisms cause specific diseases. Pasteur, a French chemist, showed through a series of elegant experiments in the 1860s that fermentation and putrefaction were caused by living microbes, not by spontaneous generation. His famous swan neck flask experiments demonstrated that broth remained sterile as long as airborne microbes were kept out, demolishing the ancient idea that life could arise spontaneously from non-living matter. And Koch, a German physician, went further. He developed a rigorous set of criteria, Koch's postulates, for proving that a specific microbe causes a specific disease. You had to find the organism in every case of the disease. You had to isolate it in pure culture. You had to reproduce the disease by introducing the cultured organism into a healthy host. And you had to re-isolate it from the newly sick host. Systematic, methodical, powerful. And here's something about Koch that's worth knowing, because it tells you about the kind of person who changes science. Koch was a country doctor, a general practitioner in a small town in what is now Poland. He wasn't at a prestigious university. He didn't have fancy equipment. He was working in his spare time, using a microscope his wife had bought him as a birthday present. And with that birthday present microscope, in a makeshift laboratory in his home, he worked out the life cycle of anthrax and proved that a specific bacterium caused the disease. When he presented his findings in 1876, the scientific establishment was stunned, partly by the quality of the work, and partly by the fact that it had come from a nobody in a provincial town. Koch was also famously meticulous and famously difficult to get along with. His rivalry with Pasteur became one of the great scientific feuds of the 19th century, fueled partly by genuine intellectual disagreements, partly by French-German national antagonism in the aftermath of the Franco-Prussian War, and partly by the fact that both men had enormous egos and absolute confidence in their own methods. They sniped at each other in publications for years, and through the 1870s and 1880s, the germ theory triumphed. Koch identified the bacteria responsible for anthrax, tuberculosis, and cholera. Pasteur developed vaccines against anthrax and rabies. One by one, the great infectious diseases were being traced to specific bacteria, specific fungi, specific parasites. The invisible enemies were being named and catalogued. Medicine was being revolutionized. It must have felt, to the scientists of that era, like they were finally winning. Like the mystery of infectious disease was being solved, one organism at a time. But there was a problem, a nagging, persistent problem that wouldn't go away, because some diseases, some very important, very deadly diseases, refused to yield to Koch's postulates. The organisms responsible couldn't be found, couldn't be seen under any microscope, couldn't be grown in any culture medium. The germ theory was working beautifully for bacteria, but something else was out there, something different, something that didn't play by the same rules. And the first real clue about what that something might be came from a completely unexpected place. Not from a hospital, not from a human disease, but from a field of tobacco plants in Crimea, where the leaves were developing strange, mottled patterns of light and dark green, and nobody could figure out why. The year was 1879, and a young German agricultural chemist named Adolf Meyer had been asked to investigate this tobacco mosaic disease, as it came to be called, which was devastating tobacco crops across the Netherlands and beyond. Meyer was methodical. He tested everything he could think of. He examined the soil, the water, the growing conditions, and then he tried something simple but revealing. He took sap from a sick tobacco plant, rubbed it on the leaves of a healthy plant, and waited. The healthy plant got sick. The disease was transmissible. Something in that sap was infectious. So far, so good. That fit perfectly with germ theory. There must be a bacterium in the sap causing the disease. Meyer tried to find it. He looked under microscopes. He tried to grow it on culture plates, the standard technique Koch had pioneered for isolating bacteria. Nothing. He couldn't find any organism. He couldn't grow anything. But the sap was definitely infectious. He'd proved that. Something was there. He just couldn't see it or culture it. Mayer concluded, somewhat tentatively, that the agent must be an unusual bacterium, perhaps one that was very difficult to grow in the lab. It was a reasonable guess. It was also wrong. And this is where the story gets really interesting, because the next step was taken by a young Russian biologist named Dmitry Ivanovsky, and what he did in 1892 was beautifully simple and absolutely pivotal. Ivanovsky took the infectious sap from diseased tobacco plants and passed it through a Chamberlain filter. Now, a Chamberlain filter is a special porcelain filter with pores so tiny that bacteria cannot pass through. It was the gold standard for separating bacteria from liquids. If you filtered a liquid through a Chamberlain filter, and the filtrate, the liquid that came through, was sterile, that proved any bacteria had been trapped. Bacteriologists used this technique all the time. Ivanovsky filtered the infected sap, expecting the filtrate to be harmless. It wasn't. The filtered sap, the liquid that had passed through the bacterial filter, was still infectious. He rubbed it on healthy tobacco leaves, and they got sick. Whatever was causing tobacco mosaic disease was small enough to pass through a filter that stopped all known bacteria. It was smaller than any living thing anyone had ever identified. Think about what that means. This was 1892. The germ theory had been established for barely a decade. Scientists were still mapping the world of bacteria, still identifying which microbe caused which disease. And now here was evidence of something else entirely. An infectious agent, so small it was invisible, even by the standards of the invisible world they'd just discovered. It was like discovering that beneath the ocean floor, there was another, deeper ocean. But Ivanovsky, interestingly, didn't fully grasp what he'd found. He suggested that perhaps the bacteria were somehow passing through the filter due to some imperfection, or that the disease was caused by a toxin produced by bacteria rather than by the organisms themselves. He didn't take the conceptual leap that his experiment demanded. That leap was made by someone else, 6 years later, a Dutch microbiologist named Martinus Beijerinck. Beijerinck repeated Ivanovsky's filtration experiments in 1898, got the same results, and then went further. He did something clever. He took the filtered sap that had passed through the Chamberlain filter, used it to infect a new batch of tobacco plants, then made a fresh extract from those newly infected plants, filtered that, and showed it was still infectious. And he could repeat this process over and over. Each time, the filtrate remained infectious. This ruled out the toxin hypothesis completely. A toxin would get diluted with each passage. Each generation of infected plants would have less of it. But the infectivity wasn't diminishing. That meant whatever was in the sap was reproducing. It was multiplying inside the plant cells. It was alive, or at least it was behaving as if it were alive, replicating itself, making more of itself, passing from one generation to the next. Beijerinck called this agent a contagium vivum fluidum, a contagious living fluid. He believed it was a new form of life, something fundamentally different from bacteria, something liquid rather than particulate. He was wrong about the fluid part. Viruses turned out to be particles, not dissolved substances, but he was absolutely right about the fundamental insight. This was a new category of infectious agent, something unprecedented, something that didn't fit into any existing classification of life. And here's what's fascinating about the timing. Almost simultaneously, in 1898, Friedrich Loeffler and Paul Frosch in Germany showed that foot and mouth disease in cattle was also caused by a filterable agent, something that passed through bacterial filters and remained infectious. This was huge. because foot and mouth disease affects animals, not plants. So the filterable agents weren't just a botanical curiosity. They were a general phenomenon. They existed across the living world. And over the next few years, more and more diseases were traced to these mysterious filterable agents. Yellow fever, rabies, smallpox, polio. One by one, diseases that had resisted every attempt to identify a bacterial cause turned out to be caused by these entities that were too small to see, too small to filter out, too small to grow on culture plates. The word virus, by the way, comes from Latin, where it meant poison, or venom, or slimy liquid. It had been used loosely for centuries to refer to any infectious agent. But in the early 1900s, it began to acquire its modern, specific meaning: a filterable, submicroscopic infectious agent that could only reproduce inside living cells. That last part is critical, and Beijerinck was the first to realize it. Viruses don't grow on their own. They don't eat, they don't metabolize, they don't do anything on their own. They need a living cell, a host cell, to make more of themselves. Without a host, a virus is just an inert particle, a packet of information waiting for something to read it. And that raises a question that scientists have been arguing about for over a century, a question that sounds simple, but turns out to be deeply difficult, and it's worth spending a moment on before we go any further. The question is this: are viruses alive? And the honest answer is that it depends on what you mean by alive. If you define life as something that can reproduce, evolve, and respond to its environment, then viruses sort of qualify. They do reproduce, they do evolve, sometimes astonishingly fast. But they can't reproduce on their own. They have no metabolism. They don't maintain homeostasis. They don't grow. Outside a host cell, a virus does absolutely nothing. It just sits there, like a message in a bottle waiting to be found. The great British biologist Peter Medawar once described a virus as a piece of bad news wrapped in protein. And honestly, that's one of the best definitions anyone has ever come up with. It captures the essential nature of a virus. It's information, molecular information, packaged in a protective shell, and when that information gets into the right cell, it hijacks the cell's machinery to make copies of itself. That's it. That's the entire program. Copy me. But we're getting ahead of ourselves, because in the early 1900s, nobody knew any of this. They knew these filterable agents existed. They knew they caused disease. They knew they were very, very small. But nobody had ever seen one. Nobody knew what they were made of. Nobody understood how they reproduced. The tools to answer those questions simply didn't exist yet. And it would take decades. Decades of painstaking work, ingenious experiments, and one truly game-changing technology before the picture began to come into focus. That technology was the electron microscope, and the experiment that changed everything involved a man in a chemistry lab at the Rockefeller Institute in Princeton, New Jersey, doing something that seemed impossible, something that blurred the line between chemistry and biology in a way that made a lot of scientists deeply uncomfortable. His name was Wendell Stanley, and in 1935, he took the juice from a ton of infected tobacco plants, literally a ton, about 1,000 kilograms of diseased plant material, and through a series of careful chemical purification steps, he managed to isolate the tobacco mosaic virus in pure form. And when he looked at what he had, it was crystals. Actual crystals, like salt or sugar or quartz. He could dissolve them and recrystallize them, the same way you'd purify any chemical compound. And when he dissolved those crystals and rubbed the solution on healthy tobacco leaves, they got sick. The crystals were infectious. Now, think about why that was so shocking. Crystals are chemistry. They're what you get when identical molecules arrange themselves in repeating patterns. Crystals are the quintessence of non-living matter, and here was an infectious agent, something that could reproduce inside living cells, something that caused disease, something that behaved, in many respects, like a living organism, and it formed crystals. It was a chemical. It could sit on a shelf in a jar, inert, indefinitely, and then spring back to infectious life the moment it encountered a suitable host cell. Nothing else in biology did that. Nothing. Stanley won the Nobel Prize in Chemistry in 1946 for this work, and it rocked the scientific world. But he also got something wrong. He initially thought the virus was pure protein. It turned out that tobacco mosaic virus is actually about 95% protein and 5% RNA, ribonucleic acid. That correction came from other researchers, including Frederick Baudin and Norman Pyrie in Britain, who showed in 1936 that the virus contained nucleic acid. And that 5% of RNA turned out to be the important part, the actual genetic material, the instructions, the blueprint. The protein was just the packaging. Meanwhile, the electron microscope was finally giving scientists their first actual look at what viruses were. The first electron microscopes were developed in the early 1930s in Germany by Ernst Ruska and Max Knoll, and by the late 1930s, they were being used to image viruses. In 1939, Gustav Kausche, Edgar Pfankuch, and Helmut Ruska, Ernst Ruska's brother, published the first electron micrographs of tobacco mosaic virus. And there they were, not fluids, not dissolved substances, not vague entities, particles, rods, tiny uniform rods, about 300 nanometers long and 18 nanometers wide. For context, a nanometer is a billionth of a meter. A human hair is roughly 80,000 nanometers wide. These things were almost unimaginably small, and they were beautiful in their regularity. Every particle looked the same. Same length, same width, same shape. It was clearly a highly organized molecular structure, not a random glob of gunk. And if you've ever seen an electron micrograph of tobacco mosaic virus, you'll know what I mean about the beauty. The particles are perfectly uniform. Each one a hollow tube made of over 2,000 identical protein subunits arranged in a helical pattern, spiraling around the central strand of RNA like the steps of a spiral staircase. The precision is breathtaking. There's no randomness, no messiness. It's molecular architecture of the highest order, assembled without any blueprint, without any supervisor, without any external guidance. Just the chemical properties of the proteins causing them to spontaneously arrange themselves into this elegant structure. Self-assembly, they call it. You can actually take purified TMV coat protein and purified TMV RNA, mix them together in a test tube, and they'll spontaneously reassemble into infectious virus particles. No cells needed, no life needed, just chemistry, just physics. Just the inherent tendency of these molecules to find their lowest energy configuration, which happens to be a perfectly formed virus particle. And as electron microscopy improved through the 1940s and 1950s, the diversity of viral forms became apparent. Tobacco mosaic virus was a rod, but other viruses had completely different shapes. Some were spherical, or rather icosahedral, shaped like 20-sided dice, with that elegant geometric symmetry that shows up again and again in mathematics and architecture. Some had complex structures, like the bacteriophages, the viruses that infect bacteria, which look almost like lunar landing modules, with a geometric head, a cylindrical tail, and spindly leg-like fibers at the base. The first time you see a good electron micrograph of a bacteriophage, it's hard to believe it's a natural object. It looks engineered. It looks like something from science fiction, but it's just a virus, one that's been infecting bacteria for billions of years, shaped by evolution into that extraordinary form. And this brings us to bacteriophages, which deserve their own moment in this story, because they turned out to be far more important than anyone initially realized. Phages, as they're usually called, were discovered independently by two scientists. The English bacteriologist Frederick Twort noticed something odd in 1915. Bacterial colonies on his culture plates were sometimes developing clear, glassy patches where the bacteria had been destroyed. Something was killing them. He published a brief paper noting the phenomenon, but didn't pursue it extensively. Then, in 1917, a French-Canadian microbiologist named Félix Derel, working at the Pasteur Institute in Paris, independently discovered the same phenomenon, and he went much further. Derel was investigating an outbreak of dysentery among French soldiers, and he noticed that filtrates from the soldiers' stool samples could destroy dysentery bacteria in culture. He named the invisible agents bacteriophages, bacteria eaters, and immediately grasped their medical potential. If there were viruses that killed bacteria, couldn't you use them to treat bacterial infections? Derel was one of those larger-than-life characters that science occasionally produces. He was self-taught, restless, flamboyant. He spent time in Guatemala studying coffee blight, in Mexico fighting a locust plague, in Indochina working on cholera. He conducted some of the first phage therapy experiments, treating dysentery and cholera patients with preparations of bacteriophages, and reported remarkable successes. Phage therapy became a genuine medical movement in the 1920s and 1930s, particularly in the Soviet Union and Eastern Europe, where it was championed by the Georgian scientist George Eliava, who founded a major phage therapy institute in Tbilisi that still operates today. But in the West, phage therapy largely fell out of favor after the discovery of penicillin and other antibiotics in the 1940s. Antibiotics were easier to mass produce, more predictable, more broadly effective. Why mess with finicky viruses when you had chemical compounds that could wipe out entire classes of bacteria? It seemed like an obvious choice, and for decades it was. But we'll come back to phage therapy later, because the story has a twist that has everything to do with the antibiotic resistance crisis that's one of the most pressing public health challenges of the 21st century. For now, though, the bacteriophages had another role to play in science, a role that had nothing to do with therapy and everything to do with understanding the most basic questions in biology. Because phages, with their simple structure and rapid reproduction, turned out to be the perfect experimental tools for answering a question that was consuming biology in the middle of the 20th century. What is a gene made of? By the 1940s, scientists knew that genes existed, that they carried hereditary information, and that they were located on chromosomes. But what were they, physically? What molecule carried the genetic code? The leading candidates were protein and DNA. Most scientists bet on protein, because proteins were complex and varied. There were thousands of different proteins with different structures and functions, which seemed to provide enough information capacity to encode heredity. DNA, by contrast, seemed too simple. It was made of only 4 building blocks: the nucleotides adenine, thymine, guanine, and cytosine, and many researchers assumed it was just a structural scaffold, too boring and repetitive to carry complex information. And this is where a beautiful experiment involving bacteriophages settled the question. In 1952, Alfred Hershey and Martha Chase at Cold Spring Harbor Laboratory on Long Island designed a now classic experiment using a phage called T2, which infects the bacterium E. coli. Here's what they knew. When T2 infects a bacterium, it injects something into the cell, some material that takes over the cell and forces it to make new phages. The question was, what material gets injected, the protein or the DNA? Here's how they figured it out. They knew that protein contains sulfur, but not phosphorus, while DNA contains phosphorus, but not sulfur. So they grew phages in 2 different batches. One batch was grown in media containing radioactive sulfur, which would label the protein. The other batch was grown in media containing radioactive phosphorus, which would label the DNA. Then they let these labeled phages infect bacteria. After the phages had attached and injected their material, Hershey and Chase used a kitchen blender, literally a wearing blender, the kind you might make smoothies in, to violently shake the phage ghosts off the outside of the bacteria. Then they spun the mixture in a centrifuge to separate the heavy bacteria from the lighter phage remnants. The result was beautifully clear. The radioactive sulfur, the labeled protein, stayed outside, in the phage ghosts. The radioactive phosphorus, the labeled DNA, went inside, into the bacteria, and it was the bacteria, containing the DNA, that went on to produce new phages. DNA was the genetic material, not protein. DNA, that 4-letter molecule that everyone had thought was too simple, was, in fact, carrying the entire blueprint of life. The Hershey-Chase experiment wasn't the only evidence. Oswald Avery's work a few years earlier had pointed in the same direction, but it was definitive in a way that convinced most holdouts. And just 1 year later, in 1953, James Watson and Francis Crick, building on Rosalind Franklin's X-ray crystallography data, worked out the double helix structure of DNA, and suddenly, everything fell into place. The structure of DNA explained how it could carry information, the sequence of bases along the strand, those 4 letters, A, T, G, and C, arranged in specific orders, spelled out the instructions for building proteins, and it explained how it could be copied. The 2 strands could separate, and each could serve as a template for building a new complementary strand. It was elegant, it was powerful, and it launched the revolution in molecular biology that continues to this day. And viruses were at the center of that revolution, because their simplicity made them the ideal model systems. If you wanted to understand how genes work, how DNA is replicated, how proteins are made from genetic instructions, viruses were the perfect test subjects. They had tiny genomes, a handful of genes, compared to the thousands in bacteria or the tens of thousands in human cells. You could study them quickly, manipulate them easily, and learn fundamental principles that applied to all of life. Much of what we know about molecular biology, about how the genetic code works, about gene regulation, about how mutations arise, was first discovered using viruses, particularly bacteriophages. The phage group, a loose network of scientists, including Max Delbruck, Salvador Luria, and Alfred Hershey, pioneered this approach in the 1940s and 1950s, and they effectively created the field of molecular biology. All 3 shared the Nobel Prize in 1969 for their work on viral genetics. But now let's talk about how viruses actually work, because this is where it gets really clever, and I mean clever in an engineering sense. The mechanisms that viruses use to hijack cells are so elegant, so efficient, so perfectly designed by evolution that you almost can't help admiring them, even as you recognize the havoc they cause. Here's the basic cycle, and then we'll get into the variations, because the variations are where the real magic is. A virus particle, a virion, if you want the technical term, floats around in the environment. It's inert. It's not doing anything. It's just a package. Then it bumps into a cell. Not just any cell, a very specific type of cell, because viruses are extraordinarily picky about which cells they can infect. This pickiness comes down to surface proteins, On the outside of the virus, there are proteins that act like keys. On the surface of the host cell, there are proteins that act like locks. If the key fits the lock, the virus can attach. If it doesn't fit, nothing happens. The virus just bounces off and keeps drifting. This is why different viruses infect different tissues, different organs, different species. The cold virus targets cells in your upper respiratory tract because those cells have the right surface receptors. HIV targets a specific type of immune cell called a CD4 positive T cell because those cells display the CD4 receptor that HIV's surface protein can latch onto. Rabies virus targets nerve cells. Hepatitis viruses target liver cells. The specificity is remarkable. It's like having a key that opens only one particular door in a building with millions of rooms. Once the virus has attached, it needs to get inside, and there are several ways this can happen, depending on the virus. Some viruses fuse their outer envelope with the cell membrane and dump their genetic material directly into the cell. Some get swallowed whole by the cell through a process called endocytosis. The cell membrane wraps around the virus particle and pulls it in, like the cell is eating it. Bacteriophages do it differently. They sit on the outside of the bacterial cell and inject their DNA through the cell wall, like a syringe. The T4 phage actually contracts its tail sheath to drive the injection tube through the bacterial membrane. It's mechanical, it's physical, it's extraordinary. Once the viral genetic material is inside the cell, the takeover begins. And this is where you have to appreciate just how ruthless and efficient the viral strategy is. The virus doesn't bring its own machinery. It doesn't carry its own protein-making equipment, its own energy supply, its own raw materials. It brings only information, the instructions for making more viruses. And those instructions are written in the same genetic language the cell uses. So the cell's own machinery, its ribosomes, its enzymes, its energy systems, reads the viral instructions and follows them, obediently producing viral proteins and copying the viral genome, assembling new virus particles as if that were its normal job. The cell has been reprogrammed. Its own operating system has been hijacked and replaced with the virus's program. And then, once enough new virus particles have been assembled inside the cell, sometimes hundreds, sometimes thousands, they need to get out. Some viruses do this by budding off from the cell membrane, wrapping themselves in a little piece of the cell's own membrane as they go, which is actually where enveloped viruses get their outer coat. It's stolen cell membrane. Others are less subtle. They simply burst the cell open, lyse it, killing it in the process, and releasing a flood of new virus particles into the surrounding tissue. Each of those particles can then go on to infect another cell, and the cycle repeats. Exponential growth. One virus becomes 100. 100 become 10,000. 10,000 become 1,000,000. This is how infections spread within your body, and the speed can be staggering. But here's where it gets really interesting, because not all viruses use DNA as their genetic material. Some use RNA, and the RNA viruses have a whole different set of tricks, some of which are genuinely wild. Most cellular life on Earth uses the same information flow. DNA is transcribed into RNA, which is translated into protein. This is such a fundamental pattern that Francis Crick, one of the discoverers of DNA structure, called it the central dogma of molecular biology. DNA makes RNA, makes protein. It's the assembly line of life, and most DNA viruses follow this same pattern. They get their DNA into the cell transcribes it into RNA, and the RNA is translated into viral proteins. But RNA viruses skip the first step. Their genome is already RNA. Some of them. The positive sense RNA viruses, like coronaviruses and the common cold rhinoviruses, carry RNA that can be directly read by the cell's ribosomes as if it were a messenger RNA. The moment it gets into the cell, the ribosomes start translating it into protein. No transcription step needed. It's like handing someone a document that's already in their language. They can start reading immediately. Other RNA viruses, the negative sense ones, like Ebola and influenza, carry RNA that's the mirror image of a readable message. It has to be copied into a positive sense version first, before the cell can read it. These viruses need to bring along their own enzyme to do that copying, because the cell doesn't have the right equipment. So they pack a special RNA-dependent RNA polymerase inside the virus particle, ready to go the moment they enter a cell. And then there are the retroviruses, and this is where the central dogma gets turned completely on its head. Retroviruses carry RNA as their genome, but they don't use it directly. Instead, they carry an enzyme called reverse transcriptase that converts the RNA into DNA. Let that sink in. RNA into DNA. The information flows backward, from RNA to DNA, the reverse of the normal cellular process. Hence the name retrovirus. Retro meaning backward. Once the viral RNA has been converted into DNA, that DNA gets inserted into the host cell's own genome, integrated right into the cell's chromosomes, becoming a permanent part of the cell's genetic material. And every time that cell divides, it copies the viral DNA right along with its own. The virus has become part of the cell, part of the organism, in some cases, part of the species. The discovery of reverse transcriptase was one of the great shocks of 20th century biology. It was independently discovered in 1970 by Howard Temin at the University of Wisconsin and David Baltimore at MIT. Temin, in particular, had been arguing for years that something like reverse transcription had to exist, based on his experiments with a tumor-causing retrovirus called Rous sarcoma virus. The scientific establishment had been deeply skeptical. The central dogma was considered almost sacrosanct, and the idea that information could flow from RNA to DNA seemed heretical. Temin was sometimes ridiculed at conferences, but he was right. When he and Baltimore simultaneously published their discovery of reverse transcriptase in 1970, in back-to-back papers in the journal Nature, it was immediately recognized as a transformative finding. Both shared the Nobel Prize in 1975. And here's why reverse transcriptase matters so much, beyond virology. It became one of the most important tools in all of molecular biology. The ability to make DNA from RNA templates gave scientists the power to create what are called complementary DNA libraries, collections of DNA copies of all the RNA messages a cell is producing at any given time. This was revolutionary for studying gene expression, for cloning genes, for the entire biotechnology industry. A viral enzyme, an enzyme that exists because retroviruses evolved it to hijack cells, became one of the foundation stones of modern genetic engineering. There's a deep irony in that. One of our most powerful tools for understanding and manipulating life came from studying entities that we're still not sure are alive. Now, here's something else about RNA viruses that has enormous consequences, and it has to do with mistakes. When DNA is copied inside a cell, the enzyme that does the copying, DNA polymerase, has a proofreading function. It checks its work. If it inserts the wrong base, it backs up and fixes the error. The error rate is incredibly low, roughly 1 mistake per billion bases copied. That's why your DNA is copied so faithfully every time a cell divides. But most RNA viruses don't have this proofreading ability. Their RNA polymerases make mistakes. roughly one mistake per 10,000 bases copied, sometimes even more. That's 100,000 times more error-prone than DNA replication. And here's the thing. Viral genomes are short, typically between a few thousand and a few tens of thousands of bases. So, with an error rate of 1 in 10,000, almost every new virus particle that's produced has at least one mutation somewhere in its genome. Every single copy is slightly different from the one before it. Think about what that means. A virus population isn't a collection of identical particles. It's a swarm of variants, a cloud of slightly different genomes, all competing and evolving in real time. Virologists call this a quasi-species, a buzzing, shifting population of related but non-identical sequences. And this staggering mutation rate is why RNA viruses are so good at evolving. It's why the flu virus changes every year, why you need a new flu shot each season. It's why HIV is so hard to treat. The virus mutates so fast that drug-resistant variants can emerge within weeks. It's why coronaviruses keep producing new variants. The mutation rate isn't a flaw in the viral replication machinery. It's a feature. It's an evolutionary strategy, generating enormous genetic diversity that allows the virus population to adapt rapidly to any new challenge. New immune responses, new drugs, new hosts. And that brings us to the immune system, because understanding viruses means understanding the war they fight with your body every single day. And it is a war, in the truest sense. An arms race that's been running for hundreds of millions of years, each side evolving countermeasures to the other's weapons, each side driving the evolution of the other in an endless cycle of innovation and adaptation. Your immune system is, honestly, one of the most sophisticated defense systems in the known universe. And the way it deals with viruses is both elegant and brutal. Let's walk through it. The first line of defense isn't even your immune system in the traditional sense. It's your skin. It's the mucous membranes lining your nose and throat and lungs and gut. These are physical barriers, walls that most viruses simply can't penetrate. Your skin is covered in dead cells, layered like tiles on a roof, and most viruses can't get through intact skin. That's why respiratory viruses evolved to target the moist, delicate membranes of the respiratory tract, where the barriers are thinner and the cells are exposed. It's also why so many viruses exploit wounds, bites, or other breaches in the barrier. But when a virus does get past the outer defenses, the innate immune system kicks in. This is the rapid response force, the first responders. It's ancient. Versions of innate immunity exist in virtually every animal, and even plants have analogous systems. The innate immune system doesn't target specific viruses. It recognizes general patterns that signal danger, molecular signatures that are common to many pathogens but not found in your own cells. Viral double-stranded RNA, for instance, is a major alarm signal. Your cells don't normally produce double-stranded RNA, but many viruses do during replication. When sensors inside your cells detect double-stranded RNA, they trigger a cascade of defensive responses. One of the most important of these responses is the interferon system. When a cell detects that it's been infected by a virus, it releases signaling proteins called interferons, so named because they interfere with viral replication. These interferons don't directly kill the virus. Instead, they act as warning signals to neighboring cells. They're molecular alarm bells. Neighboring cells that receive the interferon signal go into a kind of defensive lockdown. They activate hundreds of genes that make them more resistant to viral infection. They slow down their protein-making machinery so that any virus trying to hijack them will have a harder time. And they display signals on their surface that help the immune system's killer cells find and destroy infected cells. And then there are the natural killer cells, and yes, that's their actual name. Natural killer cells, or NK cells, are part of the innate immune system, and they roam your body looking for cells that are stressed or infected. When they find one, they release toxic granules that puncture the target cell's membrane and trigger it to self-destruct. It's brutal, but effective. Better to sacrifice one infected cell than to let it keep pumping out virus particles. All of this happens fast, within hours of infection. But the innate immune system, powerful as it is, has a limitation. It's generic. It recognizes broad categories of danger, not specific threats. It treats every virus more or less the same way, and for many viral infections, you need something more targeted, something that can identify the specific virus that's attacking you, develop a custom-made response, and remember that response for years or decades in case the same virus ever comes back. That's the adaptive immune system, and this is where biology gets genuinely awe-inspiring. The adaptive immune system has two main branches: B cells, which produce antibodies, and T cells, which have several different roles. Let's start with B cells, because antibodies are probably the immune concept most people have heard of. Here's how it works. Your body contains hundreds of millions of B cells, and each one carries a unique antibody on its surface. These antibodies are Y-shaped proteins, and the tips of the Y are the business end, the variable region, which has a specific shape that can bind to a specific molecular target, called an antigen. And here's the remarkable part. The variable regions of all those millions of B cells are all different. Each one recognizes a different shape. Collectively, your B cells can recognize virtually any molecular structure the universe can throw at you, including molecules that have never existed before. Your immune system has pre-made answers to questions it hasn't been asked yet. How is this possible? Through a process of genetic shuffling that happens as B cells develop. The genes encoding the antibody variable regions are cut and recombined randomly, producing an enormous diversity of shapes. It's like having a set of interchangeable parts, heads, arms, and bases, that can be mixed and matched in billions of combinations. Most of these random antibodies will never encounter their matching antigen. They'll spend their entire lives on patrol without ever finding a target. But the ones that do find a match, the ones whose antibody shape fits some piece of the invading virus, like a key fitting a lock, those B cells get activated. When a B cell encounters its matching antigen, and this is the important part, it doesn't just recognize it. It becomes activated. And what happens next is one of the most extraordinary amplification processes in biology. The activated B cell starts dividing rapidly, producing clones of itself, all carrying the same antibody. But here's where it gets really clever. During this rapid division, the B cells undergo a process called somatic hypermutation, where the genes encoding the antibody variable region are deliberately mutated at a high rate. Most of these mutations make the antibody worse, but some make it better. A slightly tighter fit to the antigen, a slightly stronger grip, and the B cells with the best-fitting antibodies are the ones that get the strongest activation signals, the ones that survive and keep dividing. It's Darwinian evolution happening in real time, inside your lymph nodes, over the course of days. Your immune system is literally evolving a better antibody while you're fighting the infection. By the time the process is complete, the antibodies your body is producing can be hundreds or thousands of times better at binding the virus than the original antibody that first recognized it. It's breathtaking. Some of these clones become plasma cells, factories that pump out antibodies at a staggering rate, roughly 2,000 molecules per second per cell. These free-floating antibodies flood the bloodstream and tissues, binding to virus particles, neutralizing them, tagging them for destruction by other immune cells. Other clones become memory B cells, which persist in your body for years, sometimes decades, ready to mount a faster, stronger response if the same virus ever appears again. That's immunological memory, and it's the entire basis of vaccination, but we'll get to that. T cells, meanwhile, are doing something equally important but more direct. There are different types, but the two big ones are helper T cells and killer T cells. Helper T cells are the coordinators. They recognize infected cells and release chemical signals that activate other parts of the immune response, including helping B cells produce antibodies. Killer T cells, cytotoxic T lymphocytes, if you want the full name, are the assassins. They patrol your body, checking cells. When they find a cell that's displaying fragments of viral proteins on its surface, which infected cells do, through a molecular display system called MHC, the killer T cell latches on and delivers a lethal hit, triggering the infected cell to destroy itself. Again, sacrificing the one to save the many. The coordination between all these elements, innate immunity buying time while adaptive immunity ramps up, B cells producing antibodies while T cells hunt down infected cells, memory cells forming to provide lasting protection. It's a symphony. It's millions of cells communicating through chemical signals, making decisions, adapting in real time to a threat they've never seen before. And usually, it wins. Most viral infections you'll ever have, your immune system handles without you even noticing. You were infected. You fought it off. You never felt a thing. But viruses aren't passive targets. They've been evolving under the pressure of immune systems for billions of years, and they've developed an arsenal of countermeasures that's genuinely impressive in its devious creativity. Some viruses mutate their surface proteins constantly, staying one step ahead of the antibodies. That's antigenic drift, and it's why flu vaccines have to be reformulated every year. Some viruses, particularly influenza, can undergo antigenic shift, a sudden, wholesale reassortment of their genome segments when two different viral strains infect the same cell, producing a dramatically new virus that nobody's immune system recognizes. That's how pandemic flu strains emerge. It's not gradual, it's sudden, like shuffling two decks of cards together and dealing a hand nobody's ever seen. Other viruses have evolved specific proteins that actively sabotage the immune response. Some pox viruses produce proteins that mimic immune signaling molecules, confusing the immune system's communication network. Herpes viruses are masters of stealth. After the initial infection, they can retreat into a latent state, hiding inside cells with their genes mostly silent, invisible to the immune system, only to reactivate years or decades later. That's why cold sores come back. The herpes simplex virus never left. It was hiding in your nerve cells, in the trigeminal ganglion near your jaw, waiting for a moment of stress or immune weakness to reactivate. Chickenpox, which most people get as children, is caused by varicella zoster virus, another herpes virus. After the childhood infection resolves, the virus retreats into the dorsal root ganglia along your spine and can hide there for decades. When it reactivates, sometimes triggered by aging, stress, or immunosuppression, it causes shingles, a painful condition affecting the skin along the nerve pathway where the virus was hiding. Same virus, two different diseases, decades apart. The virus was in you the whole time. And the herpes virus family is enormous. There are 8 herpes viruses known to infect humans, and most adults carry several of them. Epstein-Barr virus, the cause of infectious mononucleosis, mono, the kissing disease, infects roughly 95% of the world's adult population. After the initial infection, it establishes latency in B cells and persists for life. Most people carry it with no ill effects, but in rare cases, or in immunocompromised individuals, it can contribute to certain cancers, including Burkitt lymphoma and nasopharyngeal carcinoma. A virus that most people carry harmlessly, quietly tucked inside their immune cells, occasionally causing devastating disease. The relationship between virus and host is more nuanced, more layered, more ambiguous than the simple story of infection and immunity would suggest. And some viruses have learned something particularly sneaky. They can actually incorporate host cell proteins into their own structure, disguising themselves as self. When HIV buds from an infected cell, it wraps itself in a piece of the cell's membrane, and that membrane comes studded with host proteins. So the virus particle, from the immune system's perspective, looks partly like a normal cell. It's wearing a disguise made from your own molecular wardrobe. HIV takes immune evasion to perhaps its most devastating extreme. It infects the very cells that coordinate the immune response, the CD4-positive helper T cells. It's like an invading army that specifically targets the enemy's generals. As HIV slowly destroys these cells over the course of years, the immune system gradually collapses, leaving the body vulnerable to infections that a healthy immune system would handle easily. That's AIDS, acquired immunodeficiency syndrome. Not a disease caused by HIV directly, but a state of immune collapse caused by the loss of those critical helper T cells, leaving the door open for opportunistic infections and cancers. But here's the thing. The immune system's ability to learn and remember is also our single most powerful weapon against viruses. Because if you can teach the immune system to recognize a virus before the virus ever shows up, you've essentially given it a head start, a rehearsal. You've trained the army before the war begins. And that's exactly what a vaccine does. The story of vaccination is one of the greatest stories in the history of medicine, and it starts, like so many great stories in science, with a keen observation and a willingness to act on it. The year is 1796. The place is the English countryside, in a village called Berkeley in Gloucestershire, and the observer is a country doctor named Edward Jenner. Now, to really appreciate what Jenner did, you need to understand just how terrible smallpox was. It wasn't just another disease. It was a civilization-shaping force. When Europeans arrived in the Americas, they brought smallpox with them, and it devastated indigenous populations who had never been exposed. Estimates vary, but some scholars believe that European diseases, smallpox chief among them, killed as many as 90% of the pre-Columbian population of the Americas. 90%. Entire civilizations collapsed. The Aztec Empire, the Inca Empire. Smallpox was already tearing through these populations before the Spanish conquistadors fully arrived. It wasn't guns or steel that did the most damage. It was a virus. And in Europe itself, smallpox was a constant, terrifying presence. It disfigured royalty and commoners alike. Queen Mary II of England died of it in 1694. Louis XV of France died of it in 1774. In the 18th century, it was the leading cause of blindness in Europe. The desire for protection was desperate, and that desperation drove the practice of variolation we mentioned earlier, deliberately infecting people with mild smallpox, a dangerous gamble that killed about 1 to 2% of those inoculated. But even that 1 to 2% mortality rate was considered acceptable compared to the 30% mortality of natural smallpox. That tells you everything about how feared this virus was. Now, Jenner wasn't the first person to notice that milkmaids seemed to be immune to smallpox. That was common folk knowledge in the dairying regions of England, milkmaids frequently contracted cowpox, a relatively mild disease they caught from the udders of infected cows, and once they'd had cowpox, they never seemed to get smallpox. Smallpox, by contrast, was one of the most feared diseases in history. It killed roughly 30% of those it infected. Survivors were often left with horrific scarring, blindness, or other permanent damage. In the 18th century, it was killing an estimated 400,000 people per year in Europe alone, and milkmaids, the ones who'd had cowpox, walked through epidemics untouched. Their smooth, unscarred complexions were proverbial, hence the stereotype of the beautiful milkmaid. Jenner decided to test the folk wisdom systematically. On May 14, 1796, he took material from a cowpox lesion on the hand of a milkmaid named Sarah Nelms, and he inoculated it into the arm of an 8-year-old boy named James Phipps. The boy developed a mild cowpox infection and recovered quickly. Then, about 6 weeks later, Jenner did something that by modern standards would be considered absolutely horrifying. He deliberately exposed young James to smallpox. He inoculated the boy with material from a smallpox lesion and waited. Nothing happened. The boy didn't get sick. He was immune. Jenner repeated the smallpox exposure several more times over the following months. Still nothing. The cowpox inoculation had protected the boy against smallpox. Jenner published his results in 1798, coining the term vaccination from vacca, the Latin word for cow. And while his work was controversial at first, some critics mocked the idea, some worried that injecting cow material into people would cause them to sprout bovine features. There were actually satirical cartoons published showing vaccinated people growing cow parts. The results were so dramatic, so undeniable, that vaccination spread rapidly across Europe and beyond. Within a generation, smallpox mortality was plummeting in populations that adopted vaccination. And here's the ultimate payoff of Jenner's discovery, and it's one of the greatest achievements in the history of our species. In 1967, the World Health Organization launched an intensified global campaign to eradicate smallpox. Using vaccination as their primary weapon, teams of health workers tracked down every outbreak, vaccinated everyone around it, a strategy called ring vaccination, and slowly, methodically, country by country, pushed the virus into smaller and smaller pockets. The last naturally occurring case of smallpox was diagnosed in Ali Malin, a hospital cook in Merka, Somalia, on October 26, 1977. He survived, and on May 8, 1980, the World Health Assembly officially declared smallpox eradicated, gone, the first, and so far only, human disease to be completely eliminated from the face of the earth. A virus that had killed hundreds of millions of people over the centuries, that had shaped the rise and fall of empires, that had scarred and blinded and terrorized humanity for thousands of years. Gone, because a country doctor in Gloucestershire noticed that milkmaids didn't get smallpox. But here's the thing, Jenner didn't know, and couldn't have known, why it worked. He had no concept of viruses, no understanding of the immune system, no knowledge of antibodies or T cells or immunological memory. He didn't know that cowpox virus and smallpox virus are closely related members of the same family, close enough that antibodies against one cross-react with the other, but different enough that cowpox is relatively harmless to humans, while smallpox is catastrophic. He didn't know any of the mechanism. He just observed that it worked, tested it carefully, and reported the results. And that was enough to save millions of lives. The next great leap in vaccination came from Louis Pasteur, about 80 years later. Pasteur took Jenner's principle, exposed the immune system to a harmless version of a pathogen to build immunity against the dangerous version, and generalized it. Instead of relying on natural cross-reactivity between related organisms, Pasteur figured out how to deliberately weaken pathogens in the laboratory, creating what he called attenuated strains. He developed vaccines against anthrax and rabies using this approach, and he did it with a showman's flair that Jenner never had. The rabies story is particularly dramatic. In July 1885, a 9-year-old boy named Joseph Meister was brought to Pasteur's laboratory in Paris, having been severely bitten by a rabid dog. Rabies at that time was essentially a death sentence. Once symptoms appeared, it was virtually 100% fatal. Pasteur had been developing a rabies vaccine by growing the virus in rabbit spinal cords and weakening it by drying those cords over potassium hydroxide. He'd tested it in dogs with promising results, but he'd never tried it in a human being. The decision to treat Joseph Meister was agonizing. Pasteur wasn't a physician, and if the treatment failed or caused harm, the legal and ethical consequences would have been severe. But the boy was going to die anyway. Over the course of 11 days, Pasteur administered 13 inoculations of increasingly virulent rabies material, and Joseph Meister survived. He never developed rabies. He went on to live into adulthood, eventually becoming the gatekeeper at the Pasteur Institute in Paris. And the rabies vaccine became a triumph, cementing Pasteur's legacy and the power of scientific vaccination. But the vaccine story that really captures the full drama, the human triumph and the human heartbreak, the scientific brilliance and the devastating mistakes, is the story of the polio vaccine. And it's worth spending some time on, because it has everything. A terrifying disease, a beloved president, 2 rival scientists, a catastrophic manufacturing disaster, and ultimately, one of the greatest public health victories in history. Polio, poliomyelitis, is caused by a small RNA virus that infects the nervous system. Most people who catch it have mild or no symptoms, but in a fraction of cases, the virus attacks motor neurons in the spinal cord, causing paralysis. Sometimes temporary, sometimes permanent, sometimes fatal if the muscles that control breathing are affected. In the early and mid-20th century, polio was one of the most feared diseases in America. Every summer brought a new wave of cases. Parents kept children indoors, away from swimming pools and crowds. The iron lung, a large mechanical respirator that enclosed the patient's body and used pressure changes to force air in and out of the lungs, became an iconic and terrifying symbol of the disease. Wards full of children in iron lungs. The images are haunting. Franklin D. Roosevelt contracted polio in 1921 at the age of 39 and was left paralyzed from the waist down. His experience galvanized the American public's determination to fight the disease. The National Foundation for Infantile Paralysis, later renamed the March of Dimes, was founded in 1938 to fund polio research and patient care, and it became one of the most successful public health fundraising campaigns in history. Millions of Americans contributed, often literally a dime at a time. The race to develop a polio vaccine pitted 2 very different scientists against each other. Jonas Salk, based at the University of Pittsburgh, pursued a killed virus vaccine, taking the polio virus, killing it with formaldehyde so it could no longer replicate, but leaving its surface proteins intact so the immune system could learn to recognize them. It was a conservative approach, safe in principle, since a dead virus can't cause infection. Albert Sabin, based at the University of Cincinnati, pursued a live attenuated vaccine, a weakened but still living strain of the virus that could replicate in the gut but couldn't cause disease. It was riskier, but potentially more effective, because a live vaccine mimics a natural infection more closely and can induce stronger, longer-lasting immunity, including immunity in the gut, where the virus actually enters the body. Salk's killed vaccine came first. It was tested in one of the largest clinical trials in medical history, the Francis Field Trial of 1954, involving nearly 2 million children, called the Polio Pioneers. The results, announced on April 12, 1955, were a sensation. The vaccine was safe and effective, reducing the risk of paralytic polio by 60 to 70% against type 1 poliovirus and over 90% against types 2 and 3. Church bells rang, people wept in the streets. Salk became a national hero overnight. But then came disaster. Just weeks after the vaccine was licensed, reports began emerging of children developing polio after being vaccinated. The cause was eventually traced to one manufacturer, Cutter Laboratories in Berkeley, California. Due to problems in their production process, some batches of the vaccine contained live, virulent poliovirus that hadn't been properly inactivated. Over 200,000 children received vaccines from the affected batches. 70,000 developed mild polio. 200 were left with varying degrees of paralysis. 10 died. It was one of the worst pharmaceutical disasters in American history. The Cutter incident, as it came to be called, led to sweeping reforms in vaccine regulation and manufacturing oversight. New safety protocols were established. The standards for inactivating virus were made far more stringent, and eventually, production issues were resolved, and Salk's vaccine proved to be safe and effective. But the incident left scars. It fueled vaccine hesitancy that echoes to this day, and it underscored a painful truth about public health, that even the most miraculous advances carry risks, that trust is fragile, and that manufacturing and quality control matter as much as scientific discovery. Sabin's live oral vaccine, meanwhile, was developed through the late 1950s, tested extensively in the Soviet Union, because American public health authorities were initially reluctant to conduct large trials of a live vaccine after the Cutter debacle, and eventually licensed in the United States in 1961 and 1962. The oral vaccine had key advantages. It was cheap, easy to administer, just a few drops on a sugar cube, and it induced mucosal immunity in the gut, which meant vaccinated people didn't just protect themselves, but also stopped transmitting the virus to others. It became the workhorse of the global polio eradication campaign, particularly in developing countries where the logistics of injectable vaccines were challenging. But Sabin's vaccine had its own risk, because it used a live, weakened virus in extremely rare cases, about 1 in 2.7 million doses. The weakened virus could mutate back to a virulent form and cause vaccine-derived polio. For decades, this was considered an acceptable trade-off, given the enormous burden of wild polio disease. But as wild polio was pushed to near extinction through vaccination, today, it remains endemic in only a tiny number of countries. The calculus shifted. In the United States and other wealthy nations, the Salk injectable vaccine replaced the Sabin oral vaccine for routine use, eliminating the risk of vaccine-derived cases. The global eradication effort continues, tantalizingly close to its goal, but hampered by conflict, mistrust, and logistical challenges in the last remaining pockets. And then, in the 21st century, the vaccine story took yet another turn, a turn that nobody in Jenner's time, or Pasteur's, or even Salk's could have imagined. And it started with a woman whose work was dismissed and ignored for decades before it changed the world. Her name is Katalin Karikó. She was born in Hungary in 1955, the daughter of a butcher, raised in a small town with no running water. She became fascinated by biochemistry and earned her PhD from the University of Szeged, working on RNA. In 1985, she left Hungary for the United States, emigrating with her family, with all their savings, about $1,200, according to the story, hidden inside a teddy bear because they couldn't take currency out of the country. She eventually landed at the University of Pennsylvania, where she became convinced that messenger RNA, mRNA, were. The idea was conceptually simple. Instead of injecting a weakened or killed pathogen, or a piece of a pathogen's protein, you inject the mRNA instructions for making that protein, and let the patient's own cells do the manufacturing. Your cells read the mRNA, build the protein, display it on their surfaces, and the immune system learns to recognize it. Elegant, clean, and, in theory, incredibly flexible. You could design a new vaccine just by changing the mRNA sequence. No need to grow and process whole viruses. But there was a massive problem. When scientists tried injecting synthetic mRNA into cells or animals, the immune system attacked it violently. The body recognized the foreign RNA as a danger signal. Remember, double-stranded RNA is one of the alarms the innate immune system watches for, and triggered a fierce inflammatory response that destroyed the mRNA before it could do its job. Kariko spent years trying to solve this problem, and the years were hard. She was denied grants repeatedly. She was demoted from the tenure track at Penn. At one point, she was essentially told that her research was a dead end and she should move on. But she didn't. In the early 2000s, working with immunologist Drew Weissman, Kariko made the breakthrough. They discovered that by modifying one of the chemical building blocks of the mRNA, replacing the natural base uridine with a synthetic variant called pseudouridine, they could make synthetic mRNA that slipped past the immune system's alarm sensors. The modified mRNA was read by cells just fine, producing the desired protein, but it didn't trigger the inflammatory response that had been killing the approach. They published the key paper in 2005. It was barely noticed. Almost nobody cited it. The scientific world was not interested, but two small biotech companies were paying attention. One was Moderna, founded in 2010 in Cambridge, Massachusetts, built specifically around mRNA technology. The other was BioNTech, founded in 2008 in Mainz, Germany, by a husband and wife team of immunologists, Ugur Sahin and Özlem Türeci, who were originally focused on cancer immunotherapy. Both companies licensed Kariko and Weissman's modified nucleoside technology and spent years developing mRNA delivery systems, lipid nanoparticles, tiny fat bubbles that could protect the fragile mRNA molecule and help it enter cells. And then, in January 2020, Chinese scientists published the genetic sequence of a new coronavirus that was causing a mysterious pneumonia in Wuhan. Within days, literally days, BioNTech and Moderna had designed mRNA vaccine candidates. They just needed to encode the spike protein of the new virus into their mRNA platform. No growing virus in eggs or cell cultures, no weakening or killing pathogens, just sequence design on a computer. BioNTech partnered with Pfizer. Clinical trials began at unprecedented speed, not because corners were cut on safety, but because regulatory reviews were done in parallel rather than sequentially. Because manufacturing was started before trials were complete, in a bet on success, and because there was no shortage of volunteers. The pandemic was raging, and people were desperate for a solution. The results were remarkable. Both the Pfizer-BioNTech and Moderna vaccines showed about 95% efficacy in preventing symptomatic COVID-19 in their initial trials. By December 2020, less than a year after the viral sequence was published, the first mRNA vaccines were being injected into arms. It was the fastest vaccine development in history, by a staggering margin. Previous records were measured in years. The mumps vaccine, previously the fastest ever developed, had taken about 4 years. And the speed was not despite the science, but because of it. Because of decades of quiet, unglamorous foundational work by people like Carico and Weissman, because of the mRNA platform's inherent flexibility, because of the lipid nanoparticle delivery technology, and because of the massive investment that a global emergency makes possible. Carico and Weissman shared the Nobel Prize in Physiology or Medicine in 2023. And the mRNA platform, their platform, is now being developed for vaccines against influenza, RSV, HIV, Zika, malaria, and a dozen other diseases. It's being explored for cancer immunotherapy, for autoimmune conditions, for rare genetic disorders. One technology, one set of foundational discoveries, opening doors in every direction. And it almost didn't happen, because a quiet Hungarian-born biochemist couldn't get a grant. Now, let's talk about pandemics, because the story of viruses is inseparable from the story of the pandemics they've caused. And while we've touched on some of this already, there are a few that deserve a closer look, because they've shaped the modern world in ways that are still unfolding. Start with the big one, the 1918 influenza pandemic, sometimes called the Spanish flu, though it almost certainly didn't originate in Spain. Spain just happened to have a free press during World War I that reported honestly on the outbreak, while wartime censorship in other countries suppressed news of the spreading disease. The first confirmed outbreaks were in military camps in the United States in the spring of 1918, though some evidence suggests the virus may have been circulating even earlier. Here's what made the 1918 flu so devastating. First, it was extraordinarily contagious. It was an H1N1 influenza A virus. We'll talk about what those letters mean in a moment. And it spread with terrifying speed through a world where people were packed together in military barracks, in trenches, in crowded troop ships crossing the Atlantic. Second, it killed in a way that was different from ordinary flu. Ordinary seasonal flu kills mostly the very young, the very old, and the immunocompromised. The 1918 virus had a peculiar W-shaped mortality curve. It killed the usual groups, but it also killed young, healthy adults in their 20s and 30s at disproportionately high rates. The leading theory for why is something called a cytokine storm. The virus triggered such an overwhelming immune response in people with strong, healthy immune systems that the immune system itself became destructive, flooding the lungs with fluid, causing massive inflammation, essentially drowning the patient in their own immune response. The stronger your immune system, the harder it fought, and in some cases, the harder it fought, the more damage it did. Your own defense system became the weapon that killed you. The numbers are staggering. The pandemic came in 3 waves: spring 1918, fall 1918, and winter 1918-1919. The second wave, in the fall, was by far the deadliest. People who seemed fine in the morning were dead by nightfall. Hospitals were overwhelmed. Coffin makers couldn't keep up. Mass graves were dug. Schools and theaters and churches closed. Cities imposed mask mandates and gathering bans, and there were protests against those mandates. The parallels with 2020 are almost eerie. Estimates of the total death toll have been revised upward over the years as better data has become available. Current estimates range from about 50 million to possibly as high as 100 million deaths worldwide, at a time when the global population was less than 2 billion. That's somewhere between 3 and 5% of all humans alive. In some communities, particularly indigenous communities and island populations with no prior exposure to influenza, mortality rates reached 50 or 60%. And the tragedy of 1918 was compounded by the fact that nobody knew what was causing it. Remember, Viruses hadn't been seen yet. The electron microscope was still 2 decades away. Scientists assumed the disease must be bacterial. The leading candidate was a bacterium called Bacillus influenzae, identified by Richard Pfeiffer during an earlier flu epidemic, which is why the disease was called influenza. But Bacillus influenzae wasn't always present in flu patients, and the filtration experiments that might have identified a viral cause were inconclusive in the chaos of the pandemic. It wasn't until decades later that the true cause, an influenza virus, was confirmed. In the 1990s and early 2000s, a remarkable scientific detective story unfolded when researchers led by Jeffrey Taubenberger at the Armed Forces Institute of Pathology managed to recover fragments of the 1918 virus's RNA from preserved tissue samples of soldiers who had died in the pandemic, and from the frozen remains of an Inuit woman who had been buried in permafrost in Alaska in 1918. Using these fragments, they eventually reconstructed the entire genome of the virus. It was an H1N1 influenza A virus. The H and N refer to the 2 major surface proteins, hemagglutinin and neuraminidase, which are the keys the virus uses to enter and exit host cells. The reconstructed virus was studied under the highest biosafety conditions, and researchers confirmed that it was extraordinarily virulent, far more so than typical flu strains, partly because of specific mutations in its genes that enhanced its ability to replicate in human lung tissue. The 1918 virus disappeared as mysteriously as it arrived. By the summer of 1919, the pandemic was over. The virus continued to circulate as a seasonal flu strain for decades, causing far less severe disease as the population developed immunity. But its legacy was enormous. Not just the death toll, but the lessons it taught, and the lessons that were ignored. The world largely forgot the 1918 pandemic with surprising speed. It was overshadowed by the war, by the political upheavals that followed, by a collective desire to move on. The first comprehensive history of the pandemic wasn't published until the 1970s. It became, as some historians have described it, a forgotten pandemic, until 2020 made everyone remember. And then there's HIV and AIDS. A slower pandemic, measured not in months but in decades, but no less devastating. The story begins with a virus that made the jump from chimpanzees to humans in Central Africa, probably in the early 20th century, most likely through contact with infected blood during hunting and butchering of chimpanzees. The virus circulated quietly for decades, spreading slowly through human populations, undetected. It wasn't until the early 1980s that the disease it caused, a mysterious constellation of rare infections and cancers appearing in otherwise healthy young men, was recognized in the United States. In June 1981, the CDC published a report noting unusual cases of pneumocystis pneumonia in 5 young, previously healthy gay men in Los Angeles. That report is generally considered the beginning of the AIDS epidemic, though the virus had been spreading silently for years before anyone noticed. The early years of the AIDS crisis were among the most painful chapters in modern medical history, marked by fear, stigma, and a painfully slow response from governments and the medical establishment. Because the disease initially appeared primarily in gay men, in intravenous drug users, and in Haitian immigrants, marginalized communities all, it was not treated with the urgency that a disease killing young people in the prime of their lives demanded. The Reagan administration barely acknowledged the epidemic for years. Funding for research was slow in coming, and people were dying horribly, their immune systems collapsing, their bodies ravaged by infections that healthy people never get. The average time from HIV infection to death without treatment was about 10 years. There was no cure and no effective treatment for over a decade. The identification of the virus itself involved a bitter priority dispute between Luc Montagnier's team at the Pasteur Institute in Paris and Robert Gallo's team at the National Institutes of Health in Maryland. Both groups published papers in 1983 and 1984 identifying a retrovirus, a virus using reverse transcriptase to insert its genome into host cell DNA, as the cause of AIDS. The dispute over who discovered it first dragged on for years and became entangled in national pride and personal animosity. Eventually, a diplomatic resolution credited both groups, and Montagnier shared the Nobel Prize in 2008, though the awarding of the prize to Montagnier and Françoise Barré-Sinoussi, while excluding Gallo, remains controversial in scientific circles. The breakthrough in treatment came in the mid-1990s with the development of highly active antiretroviral therapy, HAART, or what's now simply called ART, antiretroviral therapy. The key insight was that using a combination of drugs targeting different steps in the virus's replication cycle could suppress the virus to undetectable levels and prevent the emergence of drug-resistant variants. Since the mid-90s, HIV has gone from a death sentence to a manageable chronic condition for people with access to treatment. People diagnosed today and started on treatment promptly can expect a near-normal lifespan, but access is the key word. The global distribution of treatment has been deeply unequal. In sub-Saharan Africa, where the epidemic hit hardest, millions of people went without treatment for years because the drugs were prohibitively expensive. The fight to make generic antiretrovirals available in developing countries was one of the great public health battles of the early 21st century, involving pharmaceutical companies, governments, activist organizations, and international bodies. Programs like PEPFAR, the President's Emergency Plan for AIDS Relief, launched by the Bush administration in 2003, and the Global Fund to Fight AIDS, Tuberculosis, and Malaria have saved millions of lives. But HIV continues to infect roughly 1.3 million people per year worldwide, and about 40 million people are living with the virus today. A vaccine against HIV remains elusive, despite decades of effort. The virus's extraordinary mutation rate and its ability to hide in latent reservoirs inside the body's own cells make it one of the most difficult vaccine targets imaginable. And then there's Ebola, which deserves a mention because it represents a different kind of viral threat, one that's extraordinarily deadly, but, mercifully, not very good at spreading. Ebola virus disease has a case fatality rate that has ranged from 25 to 90% in different outbreaks, depending on the strain and the quality of medical care available. It kills by causing massive hemorrhaging and organ failure. But Ebola spreads only through direct contact with the bodily fluids of infected people or corpses, not through the air, which limits its reach. The largest Ebola outbreak in history occurred in West Africa from 2013 to 2016, primarily in Guinea, Sierra Leone, and Liberia, killing over 11,000 people. It overwhelmed healthcare systems that were already fragile, and the response was hampered by a slow international reaction. The outbreak was eventually contained through a combination of public health measures and, toward the end, an experimental vaccine, another triumph of virology applied under extreme pressure. And then, of course, there's the pandemic that's freshest in everyone's memory, COVID-19. Caused by SARS-CoV-2, a coronavirus, named for the crown-like spikes on its surface, corona being Latin for crown. The virus emerged in Wuhan, China, in late 2019, and within months had spread to every continent. The speed of spread was a product of the modern world: international air travel, global supply chains, dense urbanization. The virus moved at the speed of Boeing 747s, not at the speed of sailing ships or even railways. The story of the COVID-19 pandemic is one of extraordinary scientific achievement and extraordinary institutional failure, often happening simultaneously. The virus's genome was sequenced within weeks of its emergence. Diagnostic tests were developed rapidly. The fundamental biology of the virus, how it enters cells using its spike protein binding to the ACE2 receptor on human cells, how it replicates, how it evades immune responses, was worked out with unprecedented speed by a global scientific community that dropped everything else to focus on the problem. And as we discussed, mRNA vaccines were designed, tested, and deployed in under a year. But the pandemic also exposed deep fractures in public health systems, in political leadership, in social trust, in global cooperation. The early response in many countries was slow, confused, and hampered by inadequate testing capacity and insufficient supplies of personal protective equipment. Public health messages were often contradictory. Scientific uncertainty, which is normal and inevitable when dealing with a new pathogen, was weaponized by partisan actors and amplified by social media. Mask mandates, lockdowns, and vaccine requirements became flashpoints in culture wars that had little to do with epidemiology. The death toll mounted into the millions. The human cost was staggering. Over the course of the pandemic, official COVID-19 death tolls exceeded 7 million worldwide, but excess mortality studies, which compare total deaths to expected deaths, suggest the true toll was likely several times higher, perhaps 15 to 20 million or more, with the heaviest burdens falling on low- and middle-income countries with limited healthcare infrastructure. The economic disruption was equally profound. Lockdowns and restrictions cost trillions of dollars in lost economic activity. Schools closed for months, sometimes over a year, with consequences for children's education and mental health that will take years to fully assess. The pandemic laid bare inequalities that had always existed but were easy to ignore. In healthcare access, in working conditions, in the vulnerability of essential workers who couldn't stay home, in the digital divide that determined which children could attend school remotely and which simply couldn't. The emergence of variants, Alpha, Delta, Omicron, and its many subvariants, demonstrated in real time the evolutionary dynamics we talked about earlier. The virus's RNA polymerase, lacking proofreading ability, generated a constant stream of mutations. Most mutations were neutral or harmful to the virus. But occasionally, one arose that made the virus more transmissible or better at evading immune responses, and natural selection did the rest. The variant with the advantage spread faster, outcompeting the others. Evolution in action, visible over weeks and months rather than millennia. As of the mid-2020s, the acute phase of the COVID-19 pandemic has passed in most of the world. The virus continues to circulate, causing seasonal waves of infection, but population-level immunity from vaccination and prior infection has blunted its impact dramatically. However, the long-term consequences, including long COVID, a constellation of persistent symptoms affecting some people months or years after infection, and the social, economic, and educational disruptions caused by the pandemic response will be felt for years to come. And the fundamental question the pandemic raised, are we prepared for the next one, remains largely unanswered. The next pandemic pathogen is out there somewhere, probably in an animal host, accumulating the mutations it needs to make the jump to humans. It might be another coronavirus. It might be an influenza strain. It might be something nobody has heard of yet. The question isn't whether another pandemic will come, it's when. But here's where the story takes a turn that I find genuinely beautiful, because it would be a mistake to end the story of viruses on a note of fear. Viruses aren't only agents of disease. They are, in the deepest sense, agents of change. They have shaped the evolution of every living thing on this planet. They are woven into the fabric of life itself, in ways that are only now being understood. And, in the last few decades, we've begun learning to use them, not just to fight disease, but to heal. Let's start with phage therapy, because remember Darrell and his bacteria-eating viruses from the early 20th century. That story has a second act. With the rise of antibiotic resistance, one of the most serious public health threats of the 21st century, as bacteria evolve to resist the drugs we've relied on for decades, scientists have been dusting off Darrell's old idea. If you have a bacterial infection that doesn't respond to antibiotics, can you find a bacteriophage that kills that specific bacterium? The answer, increasingly, is yes. Phage therapy is experiencing a genuine revival, particularly for patients with drug-resistant infections who have exhausted all other options. There have been dramatic individual cases, patients near death from antibiotic-resistant infections who were treated with specially selected cocktails of bacteriophages and recovered. The first well-publicized modern case in the West was Tom Patterson, a professor at UC San Diego who developed a life-threatening infection with a multi-drug-resistant strain of Acinetobacter baumannii while traveling in Egypt in 2015. His wife, an infectious disease epidemiologist, organized an international effort to find phages that could attack his specific bacterium. The phages were administered intravenously, and Patterson recovered. His case helped spark renewed interest in phage therapy in Western medicine. The advantages of phages are compelling. They're highly specific. Each phage typically attacks only one species or even one strain of bacteria, which means they don't destroy the beneficial bacteria in your gut the way broad-spectrum antibiotics do. They evolve alongside their bacterial targets, potentially overcoming bacterial resistance in a way that static chemical antibiotics can't. And they're abundant. The ocean alone contains an estimated 10 to the 31st power phage particles, more phages than there are stars in the observable universe. Finding a phage that attacks a specific bacterium is usually just a matter of looking in the right place: sewage, soil, hospital wastewater. But phage therapy also has challenges. Because phages are so specific, you often need to identify the exact bacterial strain causing the infection, and then find or engineer phages that target it, which takes time. Regulatory frameworks designed for chemical drugs don't always fit biological agents that evolve and that may need to be customized for each patient. And rigorous large-scale clinical trials are still relatively few, though that's changing rapidly. Several clinical trials are underway or recently completed, and the FDA has granted compassionate use authorization for phage therapy in individual cases. The Eliava Institute in Tbilisi, Georgia, which Darrell helped establish, has been practicing phage therapy continuously since the 1930s and treats thousands of patients per year. And then there's gene therapy, using viruses not to infect, but to deliver. And this story, like so many in medicine, has both triumph and tragedy in it, and the tragedy came first. Here's the idea. If you have a genetic disease caused by a faulty gene, what if you could deliver a working copy of that gene into the affected cells? You need a delivery vehicle, something that can get into cells efficiently and release its genetic cargo. And what does that better than anything else in nature? A virus. Millions of years of evolution have made viruses exquisitely good at entering cells and delivering genetic material. So scientists figured out how to gut a virus, remove all the disease-causing genes, keep only the parts needed for entering cells and delivering a payload, and replace the viral genes with a therapeutic human gene. You take nature's best delivery system and repurpose it. You turn a pathogen into a medicine. The concept of gene therapy generated enormous excitement in the 1990s. The idea seemed so logical. If a disease is caused by a broken gene, just replace it. The first authorized gene therapy trial in the United States took place in 1990, treating a 4-year-old girl with a rare immune deficiency. But then, in September 1999, came a devastating setback. Jesse Gelsinger, an 18-year-old from Arizona with a mild form of a liver disorder called ornithine transcarbamylase deficiency, volunteered for a gene therapy trial at the University of Pennsylvania. He was injected with a modified adenovirus, carrying a corrective gene. Within hours, he developed a massive immune reaction to the viral vector itself. His organs began to fail. 4 days later, he was dead. He was the first person known to have died directly from a gene therapy procedure. Jesse Gelsinger's death was a watershed moment. It exposed problems not just with the specific viral vector used. Adenoviruses, it turned out, could trigger dangerous immune responses in some patients. But with the way the trial had been conducted, conflicts of interest emerged. Safety data had arguably been underreported. The gene therapy field went into a period of deep introspection and much more cautious, rigorous clinical development. It would take years to rebuild trust and refine the approach. The most commonly used viral vectors for gene therapy today are adeno-associated viruses, or AAVs, small, harmless viruses that can infect human cells but don't cause disease. Modified AAVs carrying therapeutic genes have been used to treat inherited blindness, a condition called Leber congenital amaurosis, where a faulty gene causes the retina's light-sensing cells to degenerate. In clinical trials, patients who had been blind or nearly blind from birth received injections of AAV carrying a working copy of the defective gene directly into their eyes, and they gained sight. Children who had never seen their parents' faces could suddenly see them. The treatment, called Luxturna, was approved by the FDA in 2017. Spinal muscular atrophy, a devastating genetic disease that destroys motor neurons in infants, can now be treated with Zolgensma, a single intravenous infusion of an AAV vector carrying a working copy of the SMN1 gene. Without treatment, babies with the most severe form of the disease typically die before their second birthday. With the gene therapy, they survive and reach developmental milestones that would otherwise be impossible. It's one of the most expensive drugs in the world, over $2 million per dose, but it's essentially a one-time cure for a previously fatal disease. And there's another viral application that's equally mind-bending: oncolytic viruses. These are viruses that selectively infect and destroy cancer cells while leaving normal cells alone. The idea has been around for over a century. Doctors occasionally noticed that cancer patients who happened to catch certain viral infections sometimes experienced temporary remissions. But engineering viruses to reliably and safely target cancer is a modern achievement. Several oncolytic virus therapies are now approved or in advanced clinical trials. Tvec, a modified herpes virus approved in 2015 for advanced melanoma, infects tumor cells, replicates inside them, bursts them open, and releases tumor antigens that stimulate an immune response against the cancer. You're essentially using a virus to teach the immune system to attack a cancer that it had previously been ignoring. And here's something else. Something that connects viruses to the deepest questions about life on Earth and about your own body. It turns out that viruses aren't just external threats that we encounter and fight off. They're part of us, literally embedded in our DNA. About 8% of the human genome, roughly the same percentage as all the protein-coding genes put together, consists of sequences derived from ancient retroviruses. These are called endogenous retroviruses, or ERVs. At various points in our evolutionary history, retroviruses infected our ancestors' reproductive cells, sperm or egg cells, and inserted their DNA into the genome. Those viral sequences were then inherited by every subsequent generation, passed down like any other gene. Over millions of years, most of these viral sequences accumulated mutations that rendered them inactive. They're fossils, molecular fossils of ancient infections. But some of them have been co-opted by the host. They've been repurposed. One of the most remarkable examples is a gene called syncytin. It's derived from the envelope protein of an ancient retrovirus, and it plays a critical role in the formation of the placenta in mammals. The same protein that an ancient virus used to fuse with host cells is now used by developing embryos to fuse cells in the placenta, creating the interface between mother and fetus. Without this viral gene, the placenta as we know it might never have evolved. Mammals might not exist in their current form. You owe your birth, in part, to a virus that infected one of your distant ancestors tens of millions of years ago. Think about that for a moment. Viruses aren't just parasites. They're not just agents of disease. They're agents of genetic innovation. They shuttle genes between organisms. They drive evolutionary change. They've contributed essential components to the machinery of life. The boundary between virus and host, between self and other, is far blurrier than anyone imagined. And here's a thought that might rearrange how you think about the living world. We tend to think of viruses as rare exceptions, as occasional invaders that show up, cause trouble, and get beaten back. But the truth is the opposite. Viruses are the most abundant biological entities on Earth. They outnumber all other forms of life combined. If you could line up all the viruses in the ocean end to end, the chain would stretch for about 200 million light-years, far beyond the nearest galaxies. And the oceans are full of them, an estimated 10 to the 31st power viral particles in the ocean, a number so large it's almost meaningless. These marine viruses, mostly phages that infect bacteria and algae, play a crucial role in the global ecosystem. They kill an estimated 20 to 40% of ocean bacteria every day, releasing the contents of those cells back into the water as dissolved organic matter. This viral lysis, this constant killing and recycling, is one of the major drivers of nutrient cycling in the ocean. Without viruses, the flow of carbon and nitrogen and phosphorus through marine ecosystems would be fundamentally different. The chemistry of the ocean would change. The atmosphere would change. The global carbon cycle, the very system that regulates Earth's climate, is partly driven by viruses killing bacteria in seawater. Viruses also drive the evolution of the organisms they infect, not just by killing them, but by transferring genes between them. This horizontal gene transfer, mediated by viruses, is one of the major forces shaping the genomes of bacteria and archaea. It's why antibiotic resistance can spread so quickly between different species of bacteria. Viruses can carry resistance genes from one species to another, like molecular postal carriers delivering packages of genetic information. So here you are, lying in the dark, and this is the picture that's taken shape over the course of our conversation tonight. Viruses are everywhere. In the air, in the water, in the soil, in every ecosystem on Earth, from the deepest ocean trenches to the upper atmosphere. They are the most abundant biological entities on the planet by an enormous margin. There are more viruses in a liter of seawater than there are people on Earth. They infect every form of life: bacteria, archaea, plants, fungi, animals, even other viruses. They've been here for billions of years, probably since the earliest days of cellular life, and they've been shaping that life from the beginning. And here's something that puts all of this in perspective. For a long time, virologists assumed they had a pretty good handle on what a virus was. Small, simple. A handful of genes wrapped in protein. But nature, as it so often does, had surprises in store. In 2003, a team of French researchers, led by Didier Raoult and Bernard Lascola at the University of the Mediterranean in Marseille, identified a new virus that shattered assumptions. They'd found it years earlier, inside an amoeba, mistaking it for a small bacterium because it was so large it was visible under an ordinary light microscope. When they finally figured out it was a virus, they were astonished. They named it mimivirus, short for mimicking microbe, and it was enormous by viral standards. Viruses are spectacularly diverse, ranging from tiny circoviruses with genomes of only about 2,000 bases and just a couple of genes, to the giant mimivirus, discovered in 2003, with a genome of over 1 million bases and more than 1,000 genes, bigger and more complex than some bacteria. The discovery of giant viruses blurred the boundary between viruses and cellular life even further, reigniting the old debate about whether viruses are alive. Some giant viruses even have their own parasites, smaller viruses called virophages that can only replicate inside cells that are already infected by the giant virus. Parasites of parasites. It's turtles all the way down, and we're still discovering them. New viruses are being identified at an accelerating rate thanks to metagenomics, the technique of sequencing all the DNA or RNA in an environmental sample and computationally identifying viral genomes within the mix. Every time someone sequences the viral community in a new environment, a hot spring, a bat cave, a human gut, a glacial ice core, they find viruses nobody has ever seen before. We've probably identified less than 1% of the viral diversity on Earth. The rest is still out there, waiting to be found. The story of viruses is really the story of life itself, viewed from an unexpected angle. It's a story about the fundamental machinery of biology: DNA, RNA, proteins, the genetic code, discovered and illuminated through the lens of these strange entities that exist at the boundary between living and non-living. It's a story about evolution, about the relentless arms race between parasites and hosts that has driven the diversification of both for billions of years. It's a story about human ingenuity, about Jenner's cowpox experiments, about Pasteur's rabies vaccine, about Salk's courage and Sabin's persistence, about Carrico's decades of quiet determination that led to mRNA vaccines that have saved millions of lives. And it's a story about humility, because for all that we've learned, we're still scratching the surface. The viral world is vast beyond comprehension, ancient beyond imagination, and woven into the fabric of life in ways we're only beginning to understand. And right now, as you lie there, your body is home to trillions of viral particles. Your virome, they call it, the community of viruses that lives in and on you, mostly phages that infect the bacteria in your gut, on your skin, in your respiratory tract. They're part of your ecosystem. They're shaping the composition of your microbiome right now, killing some bacteria, sparing others, influencing which species thrive and which don't. And buried in your DNA, those ancient endogenous retroviruses, 8% of your genome, molecular echoes of infections that happened millions of years ago. They're there too, some of them still doing useful work, most of them silent passengers, all of them part of what makes you, you. The universe of viruses is strange and wonderful. They're the simplest things that evolution has produced, and yet they've had the most profound impact on the living world. They challenge our definitions of life, of self, of the boundary between organism and environment. They've caused immeasurable suffering, and they've driven innovations that have saved countless lives. They've been our deadliest enemies, and, increasingly, our most powerful tools. And they've been doing all of this, quietly, invisibly, since long before any eye evolved to see them, since long before any mind evolved to wonder about them. So let that settle over you. Let the names and the dates and the details blur. What remains is something simple. The world is more complex, more interconnected, more deeply layered than it appears. There are invisible forces all around you, shaping the living world in ways that took centuries to even begin to understand. And the story of how we came to understand them is one of the great human adventures. You've been part of that story tonight, and wherever sleep finds you, whether you made it this far or drifted off somewhere around the milkmaid and the cowpox, the viruses are still there, doing what they've always done, infecting, replicating, evolving, driving the great, slow engine of life forward. And your immune system is there too, standing guard, doing its ancient, magnificent work. You are the product of billions of years of this arms race, refined and tested and shaped by it, and you are, in every meaningful sense, a survivor. Your ancestors survived every pandemic, every plague, every viral challenge that evolution threw at them, all the way back to the first cells in the primordial ocean. That's quite a lineage. That's quite a thing to carry into sleep. Rest well. The ancient contest between life and its smallest challengers will continue quietly through the night, as it always has, as it always will.